Okay, so today we are going to start chapter 6, which is about DNA and DNA replication. So it's a little bit of a technical chapter, but it can be pretty interesting. And I'll do my best to make it understandable for you. And we have several videos to watch as well, which will help a lot. I like to use videos because they help illustrate points very well. Okay, now I have to remember how to share my screen. Are there any questions before I get started? So homework's going well. Everything is going well for everyone. No problems. The links to our Zoom lectures are up. You can find them in the module section. And those are links to YouTube. I've made sure that all the videos are public. So you should have access. If you try and you're still having trouble, please let me know so I can try to figure out what's going on. But it should be current as of our lecture on Monday is like 95% uploaded. So it should be ready to go very shortly. And I will convert this file and upload it today from our lecture today. I am thinking of adding a Zoom lecture on Friday. How do you guys feel about that? Because you have an exam early next week. I thought we could finish chapter 6 together in Zoom and then do review and possibly get started on the next chapter next week. Are people able to join for a Zoom lecture on Friday? Please let me know. Everybody okay with that? Okay. And are you able to hear me? Anybody? Okay, we're quiet today. All right, I'm going to assume that you can hear me. And we're going to go ahead and get started with chapter 6. And grab the book real quick. It's always nice to kind of refer to that as we're going along, especially if you like to look at figures or keep track of certain figures. Okay, so DNA starts on page 114 in the textbook. All right, so this chapter is all about DNA. Okay, DNA is the molecule that holds instructions for all living things. It is the universal code for producing proteins, essentially. So DNA is short for deoxyribonucleic acid. So we just call it DNA for short instead of having to say that whole long word a lot, deoxyribonucleic acid. So a DNA molecule is a double helix. And so a helix, you just think it's basically like a coil. So it's two coils together. And it has long strands. It makes up long strings of, has two strings. And they're made up of nucleotides. So we'll talk about nucleotides on the next slide. But you can kind of think of a double helix like a rope ladder. And if you twisted the rope ladder, you would get this twist in it, a coil. And that's essentially what DNA looks like. You have two strands together that are coiling. And they're held together in the middle by these nucleotides. Okay, so if we look at this picture here, which is on page 115, so this is just showing you what each nucleotide looks like if you split open the DNA and we were just looking at one strand of the DNA. A subunit or a nucleic acid consists of three things, okay? A phosphate group, right here, a nitrogenous base, and a sugar. That's literally what the name means, deoxyribonucleic acid. So deoxy is referring to the sugar. Ribo is referring to the base, and then you have that phosphate backbone. So deoxyribonucleic acid. So always remember the three components: phosphate, sugar, and base is the component of a single nucleotide. Okay, there are 4 nucleotides that we associate with DNA, and they have very specific pairing rules. So adenine only binds with thymine. So we abbreviate adenine with A, thymine with T. And what it looks like is with adenine and thymine always bond together, you get a double bond. So there's a bond there, and there's a bond there. So it's a double bond. Guanine and cytosine are the other 2 nucleic acids, and they only bind with each other. And they have, note here, 3 bonds. It's a triple bond. These are hydrogen bonds, but it's just like if you had one piece of thread, it's easy to break. But if you have a whole bundle of thread, it's very difficult to break. So if you have a whole bunch of hydrogen bonds within the DNA, it's going to be hard to break it apart. So it's fairly strong. Okay, each strand of DNA is complementary. And so I'll show you what that means on the next slide. So remembering our base pairing rules, that adenine only binds with thymine and cytosine only binds with guanine. It's a double strand, so double helix. Adenine only binds with thymine and guanine only binds with cytosine. So if you know 1/2 of the strand, if you unzipped it and you had those nucleic bases just sticking out, you would be able to make the complementary strand because you already have a template. Okay, so a really important theme to remember about the DNA strands is that they are complementary. So as I said, if you know 1/2 of the DNA molecule, you can build the other 1/2. So for example, if this is, I'm drawing 1/2 of the DNA molecule that's unzipped. Okay, if we know that 1/2 of the strand, we know what binds to each other. So adenine to thymine, cytosine to guanine. We can determine what the complementary base pair would be. So what binds to adenine? Anybody? Thymine binds to adenine. Adenine binds to thymine. Guanine binds to cytosine, and so on. So you can determine the other 1/2 of that DNA strand because they're complementary base pairs. So remembering the whole fact that it's complementary is really important. So in order to replicate the DNA molecule, it basically unzips. I love this picture. I found it. I love it. So it basically unzips, so you have those 2 complementary those 2 strands that are unzipped, they're complementary to each other, and then you're able to replicate them. So each of those strands, after you've unzipped them, serves as a template strand for a new DNA strand. And you just follow the base pairing rules. So here's a great picture showing DNA unzipped, and this is on page 116. Really great picture. So if you unzip the DNA molecule, your nucleotide bases are open. So they serve as a template for a new DNA strand to form on both sides, okay? And so what ends up is that your 2 new DNA molecules each have 1/2 a strand from the parent DNA molecule. So because of this, it's called semi-conservative. Okay, so some facts about DNA. So DNA codes for all the information you need using just 4 nucleotide bases, those building blocks: adenine, thymine, cytosine, and guanine. All of the DNA molecules in your body, if you put them end to end, would reach to the earth, to the sun and back over 600 times. That is a lot, a lot, a lot. Okay, so if you were typing 60 words per minute, 8 hours a day, every day, it would take about 50 years for someone to type what the human genome is, which is a very long time. The human genome, or the DNA sequences in our cells, is very long, very complex. And if you can imagine just lining up the DNA, goes to the earth, from the earth to the sun and back 600 times. And that's all in one tiny cell in your body, and you have trillions of cells. So it's amazingly complex, very concentrated, and pretty cool. So another fun fact, humans and bananas share about 50% of their DNA in common. So we're 50% the same to bananas. Okay. DNA directs the production of proteins through RNA, which is an intermediate molecule that we're going to learn a little bit about. So RNA is also a ribonucleic acid, just like DNA. But the sugar backbone is different. So RNA stands for ribonucleic acid. There are 3 major differences in RNA versus DNA. RNA is one single strand instead of a double strand of DNA. The sugar in ribonucleic acid is ribose instead of deoxy, which is in DNA. And in addition, thymine is not used in RNA. Uracil replaces thiamine. So RNA is single-stranded, it's ribose sugar, and you use uracil instead of thymine. So there is substitution that has to occur. So I have a short video that goes over the difference between DNA and RNA. So we're going to watch that together. Hopefully this shows on the screen. Bear with me for a second. Are you able to see the YouTube screen?
I lost 20 pounds.
Okay, that didn't work. Give me a sec.
This is when I first discovered metabolic.
Okay, let me go to the new screen. It will take me just a second to get back to the right. Oh my goodness, sorry guys. Just bear with me for a second. My link didn't work. Sorry. Okay. We will get there. There it is. Finally. So sorry for the video, just 1 second.
it feels like DNA gets all the credit. Yes, the structure of DNA is amazing. It has a beautiful double helix structure. It has its own emoji. DNA stores genetic information and codes for your traits. However, sometimes what gets left out is how important RNA is. Without RNA, you actually couldn't get that genetic message out to your cells so that they can start producing proteins. We talk about this in protein synthesis. RNA is a very important biomolecule, just as important as DNA. In fact, RNA is even hypothesized as coming first, before DNA, in the RNA world hypothesis, something we need to make a separate video about. So let's compare and contrast DNA with RNA. First, you will find DNA and RNA in all living organisms. In eukaryotic cells, DNA tends to be found in the nucleus, while you can find RNA both inside and outside of the nucleus. Prokaryotic cells don't have a nucleus. Both DNA and RNA are nucleic acids, which are a type of biomolecule. Nucleic acids have a monomer, which, if you remember from our biomolecules video, a monomer is a building block. The monomer for nucleic acids is a nucleotide, so both DNA and RNA have nucleotides. The nucleotides of both RNA and DNA have 3 parts: a phosphate, sugar, and a base. Let's draw 6 nucleotides of DNA and 6 nucleotides of RNA. DNA is generally double-stranded, and if focusing on these 2 strands of nucleotides here, you can see they run anti-parallel to each other. RNA is generally single-stranded, so you are just seeing one strand here. The sugar in DNA is deoxyribose, and the sugar in RNA is ribose. This makes sense because DNA stands for deoxyribonucleic acid. That's helpful to know because the deoxyribose is a sugar, and nucleic acid is that type of biomolecule it is. RNA stands for ribonucleic acid, as its sugar is ribose. The bases in DNA are adenine, thymine, guanine, and cytosine. It helps to remember the popular mnemonic apples in the tree, that helps you remember that A goes with T, and car in the garage, so C goes with G, to understand how DNA bases pair. The bases in RNA are adenine, uracil, guanine, and cytosine. Notice the different one? It's uracil. So you have to change that popular mnemonic device here. Instead of apples in the tree, maybe the apples are under. Car in the garage still works though. We mentioned earlier that DNA codes for your traits, but it couldn't do that without RNA's help. In our protein synthesis video, we talk about 3 different types of RNA and their very important roles. mRNA, which stands for messenger RNA, mRNA's job is to carry a message based off of the DNA. In eukaryotic cells, DNA generally stays in the nucleus, but mRNA has the ability to leave the nucleus to take the message to a ribosome. Ribosomes make protein, and RNA is actually a major component of ribosomes. This type of RNA is called rRNA, which stands for ribosomal RNA. Finally, we discussed transfer RNA, or abbreviated tRNA. Its job is to transfer amino acids to match the correct mRNA codon. Codon charts using mRNA codons have been developed so that you can actually see which amino acid is brought for each mRNA codon. When those amino acids are joined together, they make a polypeptide chain. Proteins are made of one or more of these polypeptide chains, and proteins have tons of different roles, but we don't want to spoil it. Check out more in our protein synthesis video. Before we go, let's try a little 3-question quiz, shall we? Just pause it after the question so you have time to think about it. Question 1. If I have 8 DNA nucleotides, how many DNA bases do I have?
Okay, guys, can you answer that? If you have 8 nucleotides, how many bases do you have? Okay.
And also, how many base pairs? The answer: each nucleotide, regardless of whether it's a DNA or RNA nucleotide, has a phosphate, sugar, and a base. So 8 DNA nucleotides would have 8 bases. DNA bases pair like this, and that's 4 DNA base pairs. Question 2. If one strand of DNA has these bases shown here, ATT, GAC, Can you complete what the complementary DNA bases would be for the other DNA strand?
All right, take a second and match up the base pairs for DNA. So adenine and thymine go together, cytosine and guanine go together. Apple in the tree, car in the garage. Honey, I'm lecturing right now. Sorry about that. Okay.
The answer. So remember those base pairing rules for DNA and the popular mnemonic. If placing the bases in this image, reading in the direction shown on this image, they'd be TAA, CTG. Question 3. In a process known as transcription, a complementary RNA strand called messenger RNA has to complement the DNA. More about that in our protein synthesis video. So if I still had the original portion of DNA here, what would be the complementary RNA bases? The answer.
Oh, I paused too late.
So remember, it's asking for RNA. No thymine here, it's uracil instead. Apples under helps you remember A with U. Car in the garage helps you remember C with G. So in this portion, reading in the direction shown on this image, we'd have these RNA bases. Notice the answer is similar to the last answer, but there are uracil bases here instead of thymine. One last thing. Our models here, they're unable to show the beautiful 3D shape, the exact numbers of bases per turn, the chirality, but we've left you some links to explore because the goal of our videos is always to introduce the topic so that you can discover all the fascinating details and exceptions. Check out our further reading description to keep that curiosity going.
Okay. Let's go back to our lecture. We lost our spot. All right, there we go. Okay, so hopefully that was a useful little video showing you the differences between DNA and RNA, double-stranded versus single-stranded, different sugar, and the difference between thymine in DNA and uracil in RNA. Those are important for you to know. Those will come up on quizzes and exams. Okay, so DNA holds information on how to produce proteins. It is the molecule of heredity because it directs the production of all of the protein synthesis in your body. So what happens is DNA first directs the production of RNA, which happens in the nucleus, and then that RNA leaves the nucleus and goes on to engage with protein synthesis. Proteins are then, once they're made, they perform the majority of the cell functions and control physical traits that we see, like eye color, hair color, the shape of your nose, whatever it may be. So starts with DNA, copy DNA into RNA, and RNA is then converted during protein synthesis, or protein synthesis uses RNA to make proteins. Okay, so this picture here, I don't even know where it is. We may not have it in the book. Oh, it is. Okay, on page 120. It's a great little summary picture for you to look at. And so genetic information flows from DNA to RNA to protein in two major steps. You must know what these steps are. You have transcription and translation. Transcription happens in the nucleus. Translation is in the cytosol. One of the major differences. Transcription, you are copying the language of DNA into RNA. That is the translation from DNA, language of DNA, into the language of RNA. Then that mRNA that is formed leaves the nucleus and then engages in translation. It meets up with ribosomes and tRNAs and produces proteins in the translation process. Okay, so first, transcription. Transcription basically is If you think about it, transcribing is from one language to another. So this is where we're rewriting the DNA language into the RNA language. Okay, so it's making RNA. It follows the base pairing rules except for the fact that this is RNA, so you are using uracil instead of thymine. Okay, the molecule that results from transcription is called messenger RNA, or mRNA. There are different types of RNA that we will talk about. There's mRNA, there's tRNA, and rRNA. Okay, so mRNA is the coding sequence. tRNA we'll talk about later. We may not talk about it today. And then rRNA is ribosomal RNA. Okay, so in translation, after you have made your mRNA molecule in the nucleus through transcription, it leaves the nucleus, enters into the cytoplasm, and then the mRNA is there, it's translated into amino acid sequence, which builds a protein. So we'll talk a little bit about translation and a codon. So first of all, understanding what a codon is really important. So if we look at this up here, this illustration of an mRNA, a sequence of 3 nucleotide bases is called a codon. One codon codes for 1 amino acid. So a set of 3 nucleotide bases codes for 1 amino acid. That is a codon. So in transcription, you are making, so we're moving back to transcription again real quick. You are making an RNA molecule from DNA. So what happens during transcription is that DNA molecule has to unzip to show those base pairs, those complementary base pairs. One strand of the DNA that's unzipped is used to make a molecule of RNA, and you're substituting uracil for thymine while you're doing that. The RNA is processed to become messenger RNA, mRNA, and then, so let's just put a little M here, then it leaves the nucleus, goes into the cytoplasm through the nuclear pore, and then you have protein synthesis occur in translation, sorry. Okay, so what is a gene? There is really no simple definition for a gene. Is it a stretch of DNA? Does DNA produce a protein? What if the same protein is made from 2 different genes, which does happen? So a gene may not just be a single sequence in this case. So as we study the genome, the human genome, it actually becomes harder to come up with a single definition of a gene. But for now, for this book, for this class, we are defining a gene as a discrete unit of hereditary information that's made up of a nucleotide sequence in DNA. So a set nucleotide sequence codes for hereditary information, a discrete unit of hereditary information. Okay, so thinking about DNA. We've heard about this a lot, about getting DNA testing. Would you get a personal DNA test if you could? They do diagnostic testing and screening for cancer, for example. Would you get a DNA test to screen for cancer? What do you think? So while you think about that, for example, the BRCA1 and BRCA2 gene, which is well known for causing breast cancer and ovarian cancer, you can do a DNA test for that and find out if you're a carrier of that gene. If you're a carrier of that gene, then you are able to make decisions about how you want to manage your risks of DNA. I mean, your risks of cancer. I will tell you, I have gotten the DNA BRCA test to test for breast cancer because it runs in my family. And I am negative, which is good news. But I put off doing that test for years, but I felt it was better to know what I was up against than not know. So that's my example of getting a personal DNA test. Would you get a DNA test before you had a baby? Would you test for genetic disorders and mutations that could potentially impact your offspring? Would you do that? If you're planning to have a child, would you get a DNA test? Okay, one step further. Should we run DNA tests on newborn babies? What do you think? Things to think about, because there are implications to all of this. Okay, talking about translation. So this is the part that occurs in the cytoplasm, and it uses those 3 types of RNA that I brought up earlier. So translating that mRNA molecule that was made in the nucleus in transcription, you're translating that into protein. That takes place in the cytoplasm with the help of ribosomes. Always happens with ribosomes. So ribosomes are directly involved with protein synthesis. So ribosomes are made from proteins and ribosomal RNA, or rRNA. Then there's another type of RNA, transfer RNA, and these molecules carry amino acids to the ribosome during protein synthesis. Okay, this picture here has a lot of things going on. I'm trying to see if it's in your book. It does not look like this one is in your book. Okay, so within each ribosome are binding sites. So remember, ribosome is made up of ribosomal RNA and ribosomes. So within each ribosome, there are binding sites for the mRNA molecule, for that single strand. So note over here, the mRNA molecule is in that binding site. There are also binding sites for transfer RNA, tRNA. You have a P site and an A site, okay? So what happens, first we look up here, this is showing us a single codon. Remember, a codon is a set of 3 nucleotide bases that codes for one protein. And what happens is the tRNA molecule comes in, it has an anticodon made up of 3 nucleotide bases. Because remember, we're dealing with RNA, we're using uracil instead of thymine. This tRNA comes in, and each codon finds its complementary anticodon, or the anticodon finds a complementary codon. When it finds that, the tRNA molecule binds to the mRNA and to the ribosome, and then you have the transfer of protein onto the next amino acid, and you're growing a protein. And we're going to watch a video of this so you can see the process occurring. It really helps to visualize it more than just a picture. Okay, so we'll watch a video that will kind of show you this process a little bit more. Okay, so translation makes a molecule of protein using the genetic code. Translation is divided into 3 phases. You have initiation, elongation, and termination. So translation begins when 2 subunits of a ribosome assemble on an mRNA. So we're talking about our RNA and ribosome assemble onto the mRNA, then it's ready to go. After the mRNA is bound to the ribosome, tRNA molecules then bring in individual amino acids that match the codon on the mRNA. When the tRNA anticodon matches the codon on mRNA, then you have the transfer of an amino acid onto the growing protein chain. Okay, this is a long illustration, which is not in your book. So I'm going to try to go through this, and then we'll watch a video which will help illustrate this more, because it is pretty busy. So nothing can really happen until we have mRNA binding to that ribosomal subunit. Once it binds, then the large ribosomal subunit, this guy right here, binds to the small ribosomal subunit and the mRNA, and then you are able to have your first tRNA match. The second codon, if I back up here for a second, here's our second codon right here in the A binding site. This is the second binding site, which is the A site. You have another tRNA molecule that has a matching anticodon. This would be the matching anticodon that matches the codon of the mRNA. It binds. When that occurs, when that second tRNA binds, the large ribosomal subunit transfers the amino acid from the P site over to the amino acid on the A site. Okay, so now you're starting to grow an amino acid chain. The ribosomal shifts the mRNA. The tRNA that has already transferred its amino acid leaves, and the tRNA with the 2 amino acids shifts over to the P site, which leaves the A site empty again. By doing that, you make it available for the next tRNA to come in with the correct anticodon. It binds to the mRNA. Then we have that catalytic site reaction from the large ribosomal subunit transfers the chain of 2 amino acids over to the A site. And then the mRNA shifts, that tRNA leaves, and that process continues until you've reached a stop codon. So there are codons that code for literally stopping the transcription translation process. And so when you reach the stop codon, the mRNA and rRNA ribosomes disassemble, and that completed polypeptide chain is released. That's not the end of protein synthesis, but that is the end of the actual building of the amino acid chain. So translation results in a polypeptide. So as I said, the elongation of the polypeptide continues until you reach a stop codon on the mRNA. When you reach that stop codon, the ribosome breaks apart, the mRNA is released, and you are left with a strand of amino acids. It's kind of like pearls on a string, okay? This polypeptide can then be used by the cell as a functioning protein. But before that amino acid chain is functional, it has to be bent into a 3-dimensional shape. So further processing needs to occur, and that happens in the Golgi apparatus. Okay, so in summary, if we look at DNA, our original DNA strand right here, here's our complementary base pair strand for DNA, because remember, DNA is double-stranded. mRNA is going to be the complementary base pair, also using uracil instead of thymine. So I would encourage you to take a look at this picture and compare the mRNA with the original strand. So if we look at mRNA, it's AA, uracil instead of thymine, guanine, 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 adenine, guanine, uracil, uracil. So it matches, except for we're substituting uracil. tRNA is the complementary base pair to mRNA. So it's what would bind to adenine would be uracil. What would bind to uracil would be adenine, and so on. And then each set of 3 codes for a protein. That is your codon. Okay, so this illustration here shows you the different codon possibilities and the proteins or stop sequence that it makes. Okay, so looking at the polypeptide product, there is your mRNA molecule. Each codon is a set of 3. Each codon codes for a single amino acid. Okay, so your polypeptide product is literally a string of amino acids. Okay, so we have a couple of videos we're going to watch about this, and then we'll see how far we get after that. But I felt it would be helpful to watch these videos. Oh, wait, it's not going to, oh, is it screen sharing? Can you guys see the YouTube screen? Someone let me know, please.
No.
No? Okay, thank you.
You're my favorite.
Okay. Sorry, one second. My links aren't working the way I want them to. I believe it is this video that we're going to watch first.
You're my favorite granddaughter. Well, I can say that because
Okay, bear with me here. Looks like we have.
there's no one else listening.
All right, here we go.
After learning about DNA, have you ever wondered how can the DNA actually result in a trait? Let's take an example, like eye color. Yes, your DNA has the genetic information it codes for the color of your eyes. Your eye color is based on a pigment that is inside the eyes. But in order to have that pigment, you have genes, which are portions of DNA that can code for proteins, which help make that pigment. So what we're going to talk about is how your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means to make something. So protein synthesis means to make protein. And you may wonder, what's a big deal about proteins? Well, you may not realize this, but proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. You've got to make proteins. It's essential for you to live. And what is so cool is that you are making proteins right now as you sit and watch this video. It's happening in your cells. They're making proteins. So back to your DNA and its role in all of this. All of your cells have DNA. well, a few exceptions. And that DNA is in the nucleus. Some DNA is non-coding DNA. Some DNA makes up genes that are not activated. More about that in our gene regulation video, but we're going to talk about genes that are coding for active proteins. So how are we going to get the information from these genes out of the nucleus so that the cell can start producing the proteins that it needs to make? Well, let us introduce you to the amazing work of RNA. We have a video comparing and contrasting RNA and DNA. We'll be brief here in saying that RNA is a nucleic acid, like DNA, but it has a few differences. Its role in protein synthesis also is huge. Before we get into the process, please note our typical disclaimer. We tend to simplify topics, but as always, we hope that you have the desire to explore this complex, amazing process later on, to learn all about the extra information that we don't have the ability to include in this short video. In protein synthesis, we can look at two major steps. One is transcription and the other is translation. Transcription has a C in it and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. Now, transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's not only fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon, which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful. You can learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You can see on the codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there can be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid, leucine. That means their complementary tRNAs all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA, CCA. On the codon chart, you can see that codes for the amino acid proline. The complementary tRNA has the anticodon GGU. And look, there's the proline that we knew it would be carrying. The tRNA will transfer that amino acid and eventually leave where it can pick up another amino acid. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, but when the ribosome reaches it, it indicates that the protein building is finished. So the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA, so the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA. Protein folding and modification may occur, and the protein may need to be transported. This can all vary based on the protein structure and function. Another fascinating topic for another Amoeba Sisters video. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay. So I like that video. It's a nice, simple, whoa, my screen working right now. Nice, simple summary. Let's go back to where we were. Okay, there is one more video. Yeah, we'll watch it because it's before we start talking about gene regulation. So let me see if I can find that. Let's see, new share. I'm getting this, guys. I'm getting it. Let's see. All right. And this is from my favorite Crash Course guy. So it's the same topic. He just has a different way to talk about it.
Finger on the A key of my keyboard, but
If I can get this to start over instead of watching it from the middle. Okay.
not watching on your cell phone. It doesn't work on cell phones. I apologize for that. Thank you to everyone who helped us put this show together, and thank you to you for watching it today. If you have any questions about this episode,
Okay,
please
sorry,
leave them
just
in
a
the comments
second.
below, or you can get us on Facebook or Twitter.
It's acting up. There we go.
Where any language more than 189,000 letters. If you were to write it down, though I don't know why you would, it would fill up more than 100 pages. And if you could actually say it without like breaking your face, it would take about 5 hours. What the frick is this word? Well, it is the name of the longest known protein on Earth, and it's actually in you right now. Because of its enormous size, it was given the name Titan by scientists, and that's with 2 Y's, and it's a protein that helps give something like the springiness to your muscles. Today, we're going to be talking about DNA and how it, along with 3 versions of its cousin RNA, unleash chemical kung fu to synthesize proteins just like this. So this is going to take a while to explain. So how about we make ourselves some Hot Pockets? of my favorite ham and cheese. Every time I take a bite, I wonder, how do they do it? How do they pack exactly the same flavor? The DNA instructions and biological molecules we've been talking about. Today, that's what I'm going to do. Not actually make Hot Pockets or a person, but I'm going to be talking about DNA transcription and translation, which is how we get made into the delicious things that we are today, though hopefully none of us know how delicious people are. Animals, plants, and also Hot Pockets really are nothing more than salty water, carbohydrates, fats, and proteins combined in precise proportions following very explicit instructions. Let's say I want to make my own Hot Pocket. I would have to, one, break into the lair of the Hot Pocket company holding the secret manual. Two, read the instructions on how to make the machinery to produce the Hot Pocket and the proportions of the ingredients. Three, quickly write down that information in shorthand before I get caught by the Hot Pocket police. Four, go home, follow the instructions, build the machinery, and mix the ingredients together until I have a perfect Hot Pocket. And that is how we get us. Very simply, inside the cell's nucleus, DNA instruction manual was copied gene by transcription onto a kind of RNA and then taken out of the layer where the instructions are followed by the process of translation to assemble amino acid strings into polypeptides or proteins that make up all kinds of stuff from this titan down here to the keratin in my hair. But most of the polypeptides that get made aren't structural proteins like hair, they're enzymes which go on to act like the assembly machinery, breaking down and building and combining carbohydrates and lipids and proteins that make up variations of cell material. So enzymes are just like whatever ingenious machinery they use the factory to make this. Okay, let's start out in the layer. The nucleus. The length of DNA that we're going to be transcribing onto an RNA molecule is called our transcription unit. And let's say, in today's example, that it's going to include the gene that transcribes for our friend Titan, which in humans, at least, occurs on chromosome 2. Now, each transcription unit has a sequence just above it on the strand, and that's called upstream. Biologists call that upstream on the strand, and that sequence sort of defines when the transcription unit is going to begin. This special sequence is the promoter, and it almost always contains a sequence of 2 of the 4 nitrogenous bases that we talked about in our last episode: adenine, thymine, cytosine, and guanine. Specifically, the promoter is a really simple repetition. We got thymine, adenine, thymine, adenine, and then A, and then on the other side, A, T. Because you know how this works, right? This is called the Tata box. It's nearly universal, and it helps our enzyme figure out where to bind to the strand. Now, you'll remember from our episode about DNA structure that DNA strands run in one of 2 directions, depending on which end of the strand is free and which end has a phosphate bond. One direction is 5' to 3', and the other direction is 3' to 5'. In this case, upstream means toward the 3' end, and downstream means toward the 5' end. So the first enzyme in this process is RNA polymerase, and it copies the DNA sequence downstream of the Tata box. That's towards the 5' end, and copies it into a similar type of language, messenger RNA. Quick aside, so you'll notice that to read the DNA in order to make enzymes, we need an enzyme in the first place. So it kind of gets chicken egg here. We need the enzyme to make the DNA and the DNA to make the enzyme. So where did RNA polymerase come from in the first place if we haven't made it yet? What an excellent question. It turns out that all of these basic necessities get handed down from your mom. She packed quite a lot more into her egg than just her DNA, so we had a healthy start. So thanks, Mom. So the RNA polymerase binds to the DNA at that Tata box and begins to unzip the double helix. Working along the DNA chain, the enzyme reads the nitrogenous bases, those are the letters, and helps the RNA version of those nitrogenous bases floating around in the nucleus to find their match. Now, you might also recall from our previous episodes that nitrogenous bases only have one counterpart that they can bond with, but RNA, which is the pink one here, doesn't have thymine like DNA does, which is the green and the blue. Instead, it has uracil. So U appears here in T's place as a partner to adenine. As it moves, the RNA polymerase rezips the DNA behind it and lets our new strand of messenger RNA peel away. Eventually, the RNA polymerase reaches another sequence downstream called a termination signal that triggers it to pull off. Now, some finishing touches before this info can safely leave the layer. First, a special type of guanine is added to the 5' end. That's the first part of the mRNA that we copied, and that's called the 5' cap. On the other end, it looks like I fell asleep with my finger on the A key of my keyboard, but another enzyme added about 250 adenines onto the 3' end. This is called our poly A tail. These caps on either end of the mRNA package make it easier for the mRNA to leave the nucleus. They also help protect it from degradation. recommendations from nearby passing enzymes, while also making it easier to connect with other organelles later on. But that's still not the end of it. As if to try to confuse me to protect the secret Hot Pocket recipe, the original recipe book also contains lots of extra misleading information. So just before leaving the nucleus, that extra information gets cut out of the RNA in a process called RNA splicing. And it's something like editing this video. This process is really complicated, but I just had to tell you about two of the key players because they have such cool names. One, the SNRPs, which are small nuclear ribonucleoproteins. These are a combination of RNA and proteins, and they recognize the sequences that signal the start and end of the areas to be spliced. SNRPs bunch together with a bunch of other proteins to form the spliceosome, which is what does the actual editing, as it were, breaking the junk segments down so their nitrogenous bases can be reused in DNA or RNA, and sticking together the two ends of the good stuff. That good stuff that gets spliced together, by the way, are called the exons because they will eventually be expressed. The junk that gets cut out are just the intervening segments or the introns. The material in the introns will stay in the nucleus and get recycled. So, for instance, Titan down there is thought to have hundreds of exons when it's all said and done, probably more than 360, which may be more than any other protein. And it also contains the longest intron in humans, some 17,000 base pairs long. Man, Titan, it is just a world record holder. So now that it's been protected and refined, the messenger RNA can now move out of the nucleus. Okay, so a quick review of our Hot Pocket Mission Impossible caper so far. We broke into the layer containing the instructions, we copied down those instructions in shorthand, we added some protective coatings, and then we cut out some extra notes that we didn't need, and then we escaped back out of the lair. Now I have to actually read the notes, make machinery, and assemble the ingredients. This process is called translation. So next, rewind your memory or just watch that video again to the episode about animal cells. Do you remember the rough endoplasmic reticulum? I hope you do. Those little dots on the membranes are the ribosomes. In the process, messenger RNA gets fed into a ribosome like a dollar bill into a vending machine. Ribosomes are a mixture of protein and a second kind of RNA called ribosomal RNA or rRNA. And they act together as a sort of workspace. rRNA doesn't contribute any genetic information to the process. Instead, it has binding sites that allow the incoming mRNA to interact with another special type of RNA, the third in this caper called transfer RNA or tRNA. And tRNA really might as well be called translation RNA because that's what it does. It translates from the language of nucleotides into the language of amino acids and proteins. On one end of the tRNA is an amino acid. On the other end is a specific sequence of 3 nitrogenous bases. These two ends are kind of matched to each other. Each of the 20 amino acids that we have in our body has its own sequence at the end. So if the tRNA has the amino acid methionine on one end, for instance, it can have UAC as the nucleotide sequence on the other end. Now it's just like building a puzzle. The mRNA slides through the ribosome reads the mRNA 3 letters at a time, each set called a triplet codon. The ribosome then finds the matching piece of the puzzle, a tRNA with 3 bases that will pair with the codon sequence. That end of the tRNA, by the way, it's called the anticodon. Sorry for all the terminology. You need to know it. And of course, by bringing in the matching tRNA, the ribosome is also bringing in whatever amino acid is on that tRNA. Okay, so starting at the 5' end of the mRNA that's fed into the ribosome, after the 5' cap, for almost every gene, you find the nucleotide sequence AUG on the mRNA. The ribosome finds a tRNA with the anticodon UAC, and on the other end of that tRNA is methionine. The mRNA, like a mile-long dollar bill, keeps sliding into the ribosome so that the next codon can be read, and another tRNA molecule with the right anticodon binds on. If the codon is UUA, then the matching tRNA has an AAU on one end and a leucine on the other. And if the mRNA has an AGA, then the matching tRNA has a UCU on one end and an arginine on the other. In each case, that new amino acid gets connected on the previous amino acid, starting a polypeptide chain, which is the beginning, very beginning of a protein. But it turns out that there are lots of different ways to read this code, because UUA is not the only triplet that codes for leucine. UUG does too. And arginine is coded for by 6 different triplets. This is actually a good thing. It means we can make a few errors in copying and transcribing and translating DNA, and we won't necessarily change the end product. The process continues with the mRNA sliding in a bit more, and the ribosome bringing in another tRNA with another amino acid, and that amino acid binding to the existing chain, and on and on and on and on and on. Sometimes for thousands of amino acids to make a single polypeptide chain, for example. This whole word is basically just the names of the amino acids in the sequence and the order in which they occur in the protein, all 34,350 of them. But before we can make our own hot pockets, and that string of amino acids becomes my muscle tissue, we have some folding to do. That's because proteins, in addition to being hella big, can also contort into very complex and downright lovely formations. One key to understanding how a protein works is to understand how it folds. Scientists have been working for decades on computer programs to try and figure out protein folding. Now, the actual sequence of amino acids in a polypeptide, what you see scrolling along down there, is called its primary structure. One amino acid covalently bonded to another, and that one to another, and to another in a single file. But some amino acids don't like to just hold hands with two others. They're a bit more promiscuous than that. The hydrogens on the main backbone of the amino acids like to sometimes form bonds on the side, hydrogen bonds, to the oxygens on amino acids a few doors down. When they do that, depending on the primary structure, they bend and fold and twist into a chain of spirals called a helix. We sometimes also find several kink strands laying parallel to one another called pleated sheets. All those hydrogen bonds and pleated sheets are what make silk strong, for instance. So in the end, our promiscuous amino acids lead to wrinkled sheets. These hydrogen bonds are what help give these polypeptides their secondary structure. But it doesn't end there. Remember the R groups that define each amino acid? Well, some of them are hydrophobic. And since the protein is in the cell, which is mostly water, all of those hydrophobic groups try to hide from the water by huddling together. And that can bend up the chain some more. Other R groups are hydrophilic, which, if nothing else, means they like to form hydrogen bonds with other hydrophilic R groups. So we get more bonding and more bending, and our single file line has now taken on a massively complex three-dimensional shape. It also explains why I can fix my bed head by wetting my hair with water. The water helps break some of those hydrogen bonds in the keratin, which relaxes its structure. That way I can comb it out, and when it dries, those bonds reform, and voila, perfect hair. All of this shape caused by bonding between R groups gives our polypeptide its tertiary structure. So now we have a massively contorted polypeptide chain, and it actually contorts very precisely. Sometimes just one chain is what makes up the whole enzyme or protein. In other proteins, like hemoglobin, several different chains come together to form a quaternary structure. So quick review of structure. Sequence is the primary structure, the backbone of hydrogen bonds forming sheets and spirals. The secondary structure, R group bonds, are tertiary in the arrangement of multiple proteins together with the quaternary structure. These polypeptides are either structural proteins, like this thing at the bottom here, which you can find in your muscle or in my hot pocket. They might also be enzymes like do stuff. They can cut up biological molecules like I do with the chef's knife. They can mix stuff, and they can put stuff together. So from that one recipe book, we get all the ingredients and all the tools necessary to make me, which is better than a hot pocket. Would you all agree? Now take your time with this stuff. Feel free to watch the episode a couple of times, because next week we're going to talk.
Okay. So we have watched a couple videos about transcription and translation, and also about the difference between DNA and RNA. We have about 10 more minutes, so we'll continue on, and then I'm going to hold a Zoom lecture on Friday so we can finish this chapter, because you have a test next week. I believe it is on Tuesday. Let me double check my calendar. Today is the 10th. I believe it's on Monday, the 15th. I could be wrong. Let's double check that real quick. Let me just pull up our class calendar really quick, because I was looking at this. Okay, so I scheduled a Zoom for Friday. Yes, and your next exam is on Monday the 15th. So we'll have a Zoom on Friday to continue on with lecture. We'll have a Zoom on Monday to review or finish up if we haven't finished. And then we'll start on the next chapter. We're really flying along. Okay. So are you able to see the PowerPoint screen right now, guys?
Yes.
Yes, thank you. My mouse isn't working. That's great. Oh my goodness, sorry guys. All right, I can't get it to work, so we'll just go slide by slide for now. We won't get our animation. That's fine. I don't know what the deal is. Okay, so we have talked about how transcription occurs and translation occurs, how proteins are made. The next step to this whole story is gene regulation. And this is turning genes on or off. So different cell types are going to express different genes. So for example, not all cells need lactase, which is an enzyme that breaks down the sugar in milk. You're only going to need that in digestive cells. So you don't have all of the genes turned on at the same time. You have genes turned on for particular areas that are needed and then turned off if they're not needed. Oh, my screen is being so annoying. I'm sorry, guys. I don't know what's going on. Okay. Give me a second here. Let's see if I can figure out what's going on. Okay. 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 Are you able to see the PowerPoint, guys? Should be able to. Okay. So, X chromosome inactivation. This is an example or an extreme example of gene regulation. So remember, with gene regulation, some genes are turned on, some genes are turned off. So in X chromosome activation or bar bodies, this can occur. And so you can see this with calico and tortoiseshell cats, for example. So in females, every cell only has one functioning X chromosome. So if you remember from what we've talked about before, we get 2 copies of our sex chromosomes, one from mom, one from dad, and that determines our sex. So you're only going to see this in females, generally speaking. So if they have 2 X chromosomes, one X chromosome is inactivated and condensed into what we call a bar body. And which X chromosome becomes inactivated is completely random from cell to cell. And this explains why we get these neat color patterns and blotches in calico and tortoiseshell cats. It's because you have these bar bodies that are created, and in different areas a different X chromosome is activated, which can determine the color. So it is really, really rare to get a male calico cat. All male calicos are sterile. Can you think of why that would be? Maybe I should make that our question for the day. That's a tough question. I want you to think about it and we'll continue on. I will make up some sort of little assignment for today's Zoom lecture for credit. So please make sure you check that. It will be due at the end of the day. Okay. So there are several points along the way from DNA, from transcription to translation to protein full production. It can all be regulated at the different stages. So you can have special transcription factors that bind to DNA that turn on the transcription process. So in the second video, he was talking about spliceosomes being involved in turning on the transcription process. So this is an example of what it's talking about. So after transcription has occurred, the RNA that's created has to be altered before it can leave the DNA. So before it leaves, where's my pen? Okay. Before RNA leaves the nucleus, after transcription, it is modified. So you add the cap and the tail, which we talked about in the video. And then you have non-coding sequences called introns that can be removed. So sections of the DNA that are non-coding is introns, is removed. The sections that, oops, I circled the wrong part. The sections that stay, the numbered sections are the ones that stays. These are the introns. The sections that stay, those are called exons. Okay, so the introns are removed and then the exons are all smushed together to get a final coding sequence. So you definitely need to know the difference between an intron and an exon. Remember, exons are expressed, so those want to stay. Okay. In translation, there are several opportunities for gene regulation. So the cell is able to control the following processes. Cell can control whether translation occurs, how proteins are modified after translation. So we talked about those different levels of modification, primary, secondary, quaternary structure, tertiary and quaternary structures. The cell can control that modification. The cell controls when proteins are broken down, so how long they're being used in the cell. If they're not to be used anymore, then they're broken down. So cell can control those following steps. Okay, cell-to-cell communication. Trying to find the page where this would be. Oh, okay, page 130. Cell-to-cell communication helps control gene expression. Okay, so multicellular life. So life with more than one cell, so this is us, multicellular life depends on cell-to-cell signaling. And molecules exit one cell, travel to another cell, and bind to the outside. This starts what we would call a signal transduction pathway. So in a signal transduction pathway, a signal from one cell regulates or turns the genes on and off in the receiving cell. So in this example of signal transduction pathway, a signal molecule from another cell binds to a receptor on the second cell's plasma membrane. That goes through signal transduction where it's passed from molecule to molecule, then affects gene regulation in the DNA itself. By affecting gene regulation, you can have the production of new mRNA, which then produces new proteins. And these new proteins can then go on to perform a specific function. So this is a good example of a signal transduction pathway would be the production of insulin. So you have cell-to-cell signaling that sends a signal that insulin needs to be released. It travels to that target cell, undergoes signal transduction pathway, gets to the nucleus, affects mRNA production for the insulin gene. That leaves the cell, you produce insulin, and then you have insulin available for the body to use. So this all started with one cell releasing a signal that affects another cell, cell-to-cell signaling, and this is signal transduction pathway. So cell-to-cell communication is very important in a developing embryo. So the whole idea with development, there is frequent cell division because you're increasing the body size, and it has to be very, very carefully coordinated. So an example, in inductive signaling, this can cause cells to change shape, to migrate, or destroy other cells. So a good example of induction signaling is in a developing embryo, the developing hand. At first, those fingers are webbed, but because of inductive signaling, it causes the cells in between the fingers to die, and that separates the fingers. Cell-to-cell communication is important. Again, homeotic genes, these are master control genes. They direct where the head is going to be and body parts are going to be and shape of body parts. So in this picture here, this is showing a homeotic gene between a normal fruit fly and a fruit fly that has an extra set of wings. So see those extra wings? That is due to a mutation in the homeotic gene. Mutation, you get an extra set of wings that are usually not functional, and you can develop, if it has other mutations, you can develop body parts in different parts of the body, different head shape. Notice how the head is very different, and the thorax even has a different shape too. All of those are affected by the homeotic gene, the master control gene. Okay, we are done with our time today. And are there any questions about today? Okay, let me go to our normal screen. Okay, so no questions for today. So that's where we're stopping. We'll continue on at Friday, 8 a.m. I'll put up a new link either today or tomorrow for our Zoom meeting on Friday. Please make sure that you do your Zoom attendance assignment for the day so you get credit for being here and credit for, for being here. And I will talk to you guys on Friday. If you have any questions between now and then, email me or text me. I'll be happy to help you. Okay. Thank you very much. I hope you all have a fantastic day.